so that you can go back and do this later if you need to. Let's see, is that the one I want? Yeah, it is. You also may have noticed that our assignment for today is a little bit different than kind of the other ones that we've had so far, in that there is no Google Form version of the daily assignment for today, just a Google Doc, but it goes along pretty similarly with some of the major stuff that we're going to talk about in our notes today. So there are these four different enzymes that help DNA replicate and copy itself. And so for each one on the homework, all I want you to do is kind of write in a brief description, like 1 or 2 sentences about what is the job of this enzyme. So if you want to have it up on another tab and kind of fill it in as we go along, that's totally fine. Or if you want to wait and come back to it after we go through the notes once and kind of write down what you learned on here, that is fine as well. So again, for each one, you just fill in the box over here what it is that the enzyme does, kind of what's its job when it comes to copying our DNA. And I'll make sure to point out when we get to each one of those 4 enzymes in our notes. All right, so DNA replication, the process by which we take a strand of DNA, make a copy of it, and then put that copy into a new cell that is being made. So in order to do that, we have to open it up, read all the bases that are inside, make new copies of those bases, and then take the 2 new DNA strands and put them 1 in each cell. So there are a couple things to know about DNA as we get into replication. And one of the biggest things that I kind of very briefly touched on last time, but is going to come up a lot this time, is the fact that DNA is something called anti-parallel, meaning that the strands line up with each other, but they go in opposite directions. So each single strand of DNA, each side of the DNA, has 2 different ends. One end is called the 5' end. That's what the little kind of apostrophe mark means. In science, we say 5'. And one end is the 3' end. Don't worry about why they're called that. You don't have to know why they're called that. You just have to know that on a single string of DNA, one end is the 5' end, one end is the 3' end. So when we say DNA is anti-parallel, what we mean is that if one side of the DNA goes from the 3' end to the 5' end, the other side of the DNA is going to go from the 5' end to the 3' end. So each side of the DNA strand, so each of your 2 strands, has a different end on its side. So here on the top side, if we have a 5' end on one side of our DNA, that means the other side has the 3' end. If we go to the bottom part of our DNA, if one side has the 3' end, the other side has the 5' end. So the 3' and 5' are not only on opposite sides of a DNA strand, but they are also on opposite sides of the actual DNA itself. So if one strand has a 3' down here, that means on the other side, the 3' is going to be in the exact opposite side. So on our left side over here, we go from 3' at the bottom to 5' at the top. On our right side DNA, we go from 3' at the top to 5' at the bottom. Again, you guys don't have to worry about what exactly the 3 and the 5 mean. It has to do with like how the oxygens are set up in the sugar and phosphate. But we don't have to worry about that. The main thing to remember about 3' and 5' is that they are opposites. They are on the opposite sides of the DNA. And so it creates this anti-parallel where one side of the DNA runs in one direction, the other side of the DNA runs in the other direction. So even though they both have the same parts on either side, their heading, if one is going this way, the other one is going this way. And then across the middle is where all of our little bases line up. Again, just as a little review here, remember it's A and T that always go together. So in this case, A and T is like our little kind of green and purple. So A and T and A are always together. And same thing with G and C, or C and G. Those are always together as well. And then they're held across the middle by our little hydrogen bonds that are connecting us through here. All right, so that's probably the biggest part of DNA that we haven't talked about yet that is important for when DNA goes to make a copy of itself. We go 3' to 5' on one side, means we have 5' to 3' on the other side. The 3' side and the 5' side are always on opposite ends of each other. Either you're on the other side of the strand, or you're on the other side on the other DNA strand. But it's always opposite, whether you're going up or across. All right, so that is our DNA strands. We have a 3' side, we have a 5' side, and each strand is going facing kind of in the opposite direction. So when we're going in and copying our DNA, obviously we're going to have to open up the middle of our DNA so that we can see what is the correct order. Because when we make a copy of the DNA, we want the new DNA to have all of the bases in the exact same order as all of the bases in our original DNA. So we're going to have to open it up and read what these bases are. So you might think that we open the DNA, read the bases, make copies of them, and then close it back up. But that is actually not how it works. Instead, DNA replication is known as semi-conservative. What this means is that we have our double-stranded DNA that's together up here at the top of this picture. It then gets opened up in order for those bases on the inside to be read and copied. But then we use those two strands that we opened up in order to start making the new sets of DNA. So what I mean by that is that each copy of the DNA is going to have one of the original strands on it and a new strand that gets built. Because remember, if we know what one side of the DNA is supposed to be, we can use those base pairing rules to figure out exactly what the other side should be. So when this DNA opens up, we just put a new strand on top of the old strand. So we completely break the old strand totally apart, and each one is the base that a new strand gets built on. So that's what we mean when we say semi-conservative. Each of the new strands, in science we call it the daughter DNA. So you have your parent DNA strand, and then you use each side of that DNA so that your two daughter DNAs, your copies that you made, one strand is from the original parent DNA, and one strand is the new DNA that you copied on top of it. So you had your original, you break it apart, and then you build a new strand on each side. And that's what semi-conservative means. So we'll talk about exactly how we build these new strands and exactly how the DNA gets open, but that's a very, very kind of general process. We slowly open up the DNA strand, and then each old strand gets a new strand built on top of it. All right, so we've got our different sides, our 5' and 3' sides going in the different directions. And we've got the DNA that we know it's going to split apart, and each new DNA strand that we make is going to have one old side and one new side that make it up. Anybody have any questions so far? Anything about the 3' and 5' stuff or the semi-conservative stuff that you want me to clarify a little bit?
No.
All right. So that is sort of our general process for how it works. Let's see where are we? There we are. So the next thing to go over is, when does this happen? So a cell is living its life. It's not just going to make new DNA for no reason. Instead, it makes new DNA when it knows it's going to be making a copy of itself. So all the cells in your body of copies are copies of the original cells that you were built out of. So when we copy DNA, it happens during a part of what's called the cell cycle, called the S phase. The cell cycle is basically just like the life cycle of our cell. We have some times where the cell is growing. We have some times where the cell is breaking apart and splitting up into different cells. And we'll talk about some of this later on in the term. But when DNA happens, it's called the S phase. So that's the main thing to remember about the cell cycle, is that, at least for right now, when it comes to DNA, we're copying it during this S phase over here. So the S stands for synthesis, which is basically just a fancy scientific way of saying building stuff or making stuff. So this S phase stands for DNA synthesis, meaning building new DNA. We have some growth stuff going on. We have some splitting of the cell going on. But when it comes to copying the DNA, that all happens during this period of the cell's life cycle. So, in order to copy the DNA and make sure that it's all copied correctly and that we open and close it all the right way, we need a bunch of different enzymes, which are special proteins that help the cell do these kind of reactions, in order to make it through each step. So each one of the 4 different enzymes that I talk about coming up is one of the enzymes on that enzyme chart that I was showing you guys earlier. That is the assignment for today. So DNA replication is a very complicated process. We're just going to kind of hit on the basics, but the cell needs lots of help from all of these enzymes that have very specific jobs to do. So the first enzyme that we're going to talk about is one of my personal favorite enzymes that's out there. It is called helicase. So helicase is kind of what starts the whole process, because helicase separates the DNA double helix. So the DNA was all lined up and twisted together. Helicase is what separates it and opens it up. So it makes something called a replication fork, because you have this DNA that was together, but then it splits apart, kind of like a fork in the road. And it's all thanks to helicase. The only reason that we're able to do this process is because the DNA gets opened up. One thing that people have kind of commented on in the past is helicase is kind of like the zipper enzyme of this process, because the DNA is kind of linked together like the teeth on a zipper, and then helicase comes along and unzips it, essentially, opens it up, and creates space for the rest of that copying over process to happen. So helicase is our starting enzyme. It breaks those weak hydrogen bonds that were holding DNA together, opens up the DNA molecule, and allows the other enzymes that come along later in order to start looking at those bases that are in there. Those A's, those G's, those T's, those C's. The only reason why we know they're there is because helicase opens the DNA and lets us see them. So we start off with this big long strand of this double-stranded DNA. Then our little buddy helicase comes along and acts like a zipper and kind of forces it apart, opens it up, makes this what's called a replication fork, and unbinds all those bases from each other. So it breaks those H bonds between the bases. So as it's going through, it's unwinding the DNA and opening up each strand. So now you have all of these bases in the DNA that are no longer attached to the other side. They're just kind of open and ready to have something else come along and read what they are. So helicase is probably the sort of most simple and straightforward to understand enzyme that we have going on in this process. It's coming in, it's opening it up, and it's unzipping that DNA so that those bases are no longer stuck together. Instead, they're kind of floating out in the open. All right, so we've got helicase that is starting us off. So you might think that now we've got the DNA open, now we're ready to start copying it. Stop. Don't think yet. That actually is not the case. So there are enzymes out there that copy DNA. They'll go along and put that new strand down right on top of the old strand. So they'll read an A and they'll put the T down, they'll read the G and they'll put the C down, so on and so forth. But unfortunately, they just can't start reading the DNA on their own. They need like a starting point. They need like how, if you ever seen like the sprints at the Olympics, the people like line up on their little blocks and then use that to go and start sprinting. the enzymes that copy DNA also need their own little starting blocks. They need these starting points in order to begin that copying process. They can't just start adding bases out of nowhere. Luckily, after helicase comes through, we have a second enzyme that comes along and puts down these little starting points that are called RNA primers. And so since it's an enzyme that puts down RNA primers, scientists thought they'd be super clever. And so they just called it, oh wait, we're not there yet. They called it Primase. And we'll talk about Primase in a second. So these RNA primers, you may notice that this is RNA, not DNA. So it's a little bit different type of nucleic acid, but we use it for these starting points for DNA replication. So like I said, once the DNA is open and the bases are exposed, our DNA copying enzymes can't just start going in and putting down new bases. They need a starting point. They need this primer to start off of. So instead of the DNA just starting to get built, instead we get this little piece of RNA that goes down, and then we can come along and fill in the rest of our bases with DNA. So they're pretty small. They're only like 3 or 4 bases long, but that's all the other enzymes need in order to get started. So it's like down here, we have our original strand of DNA from the parent DNA strand. And then we put down our little primer right here so that we can start building our DNA. In this case, we're building to the left. So we use the parent strand in order to figure out what types of bases we're supposed to put down. But we can only get this process started. We can only put down the new bases if we have this little primer, if we have this little starting point to go off of. So even though the bases are open and exposed, we still need this primer starting point in order for the rest of the DNA to be copied. So like I was saying earlier, scientists saw that this enzyme was coming in and putting primers down. So being the super creative types that scientists are, they decided to name the enzyme Primase. Sometimes you'll see it as Primase, sometimes you'll see it as RNA Primase. It's the same thing. And Primase is what puts down the RNA primers that we need to have that starting point to get DNA replication to get going. So here we have our original DNA strand, our parental DNA strand down here in the dark blue. And then Primase is this little triangular enzyme. And so it comes and it puts down a short RNA primer. So that's the first thing that has to happen once these bases are exposed. We put down our little primer right here. And then our DNA copying enzyme, it's called DNA polymerase, and we'll talk about a couple different types of it later on. But DNA polymerase comes in and can start at that RNA primer and then start building out from there. So once it has the starting point, polymerase can come along and start building out and building new DNA, matching the original DNA with some new DNA bases. So if the original had an A down here, the polymerase will put a T up here. If the original had a C down here, the polymerase will put a G up here, and it builds the DNA that way. But it has to have this RNA primer in order to start. And so we need this enzyme called Primase to put down this little starting block right here. One other important thing to note about primase and polymerase, and I'll bring it up again later on as we go through this, but notice how when the primer gets put down, it starts on the 5' side and then builds towards the 3' side. And then same thing with our DNA down here. It's building towards the 3' side. That's because that's the only direction that DNA can be built. So in order to build DNA or build RNA, you have to start on the 5' side and move towards the 3' side. We'll talk about it some more later and why it's really, really important. But I just want to start to get that kind of lodged in your brain for right now. So whenever we're making DNA, we have to start at the 5' side and go towards the 3' side. That's the only direction that our cells are able to build new DNA. So we've got our helicase, opens up our DNA strand and makes those bases in the middle not linked to each other, but instead broken apart and available. We then have our primase, which comes in, puts down our little RNA starter piece, and then we can build the rest of our DNA out from there. But we need this primer in order to start the process. And so we need the enzyme called primase in order to put down that process so that we can make the rest of the DNA. All right, I know it's a whole bunch of new terminology and new ideas that I'm throwing at you guys right now. So I want to take a quick pause right here and see if anybody is still confused about helicase or primase, if there's anything about these enzymes so far that you want me to go back and go over again or explain a little bit more.
Can you say that word again?
Which one?
ENZYEM. I mean,
This
ME.
is an enzyme.
Can you go over that more?
So enzymes are special proteins that help the cell do different jobs. In this case, with DNA replication, where we're copying the DNA, it's a very complicated process. So there are a bunch of different enzymes, a bunch of these different helper proteins that come in and help out. So the first enzyme that really gets things started is called helicase. And helicase is an enzyme that takes the twisted, wound up DNA, unwinds it, and opens it up so it splits the two sides of the DNA apart, like what's kind of going on in our little picture down here. That's our first enzyme, helicase. And then our second enzyme is called primase. And it puts down this short little segment of RNA, which is related to DNA, but a little bit different. It puts it down in order to give the other helper enzymes a starting point to build the rest of the DNA. So we have these open DNA strands, and we're going to put new DNA on top of them to match up with the original DNA. But in order to get that process started, we need this little primer on the DNA right here so that there's this kind of starting point that the DNA can build from. And in order to get our little primer put down, we need this helper enzyme called Primase that comes along and is an enzyme that puts down these little primers on top of this opened up DNA strand.
Thank you.
Yeah, no problem. Anybody else have any questions on anything? We're only going to be building up and getting more complicated from here. So if there's anything about the enzymes or the 5' 3' stuff or anything like that that you're still a little bit confused about, please ask now. All right. Sounds like we're all okay with this so far. So we have this enzyme that's been opened up, and we have this primer that's been put down. So whenever we're copying DNA, we don't just start at one end of the DNA and work our way all the way down, because DNA is huge. It's millions and millions of these bases long. So instead, there are lots of areas along the DNA that get opened up by helicase enzymes. So in this case, here we have our original strand of DNA over here and over here. And they're too small to see even on this picture, this microscope picture. But there's a little helicase enzyme right here that's going this way, and a little helicase enzyme right here that's going this way. So they started like right here and are moving in opposite directions, creating these replication forks on either side of the DNA. So remember, like I mentioned, our DNA has these kind of opposite sides to it, where we have the 5' and the 3' that are opposite from each other. So one strand is going in the 3', 5' direction, and the other strand is going in the 5', 3' direction. And again, the thing about making new DNA, building new DNA bases, is that it always has to go in the 5' to 3' direction. I'm actually going to highlight this here really quick just to really show it off. It's the classic yellow. So the only way that we can build DNA is we have to build from the 5' end towards the 3' end. That's the only way that our other DNA enzymes can put down new DNA. So because of this, we end up with a little bit of not necessarily a problem, but two different sides of the DNA and two different ways that it is being copied. We have what's called the leading strand and the lagging strand of DNA. And it's a little bit complicated how they work, but I'll try to show you guys kind of piece by piece how it goes. So on our leading strand of DNA, let's imagine that we're looking at this kind of right-hand half of our DNA over here. We have our little helicase enzyme right here, and it's going this way. It's opening up new DNA in this direction. So down here on the bottom of our DNA segment is what's called the leading strand. And the way that you can tell that it's the leading strand is because in green, we have our little RNA primer right here. So that was our starting point. And then in red, we have the new DNA strand that we're making on top of our original DNA strand. So the leading strand is easy to find because it's the one that is constantly just putting down new DNA bases as we go along. So it's this continuous addition of bases. Because again, our primer over here is starting from the 5' side. And remember, we have to go 5' to 3'. So if our 5' side is over here, our 3' side is on the opposite end over here. So that means that as long as Helicase keeps opening up our DNA, we can just keep putting down new bases in one long, easy to go straight line. So we put down our little primer, and then we can have a big, long, connected line of DNA bases. So the leading strand is like the easy side. You're constantly getting new bases opened up, and you can just put down your new bases right on top of them. So leading strand, one big long strand, easy to see, easy to understand. And it's all because we have the 5' where we started, and we're moving towards the 3' as we're going along, as we're opening up. So that one is pretty easy, straightforward. If you see one big long strand of DNA being put down, that is your leading strand. The lagging strand, unfortunately, runs into a little bit of a problem. The problem with the lagging strand is that it keeps opening at the 5' side. So what that means is that the DNA got opened a little bit. We were able to put down our primer right here, but then DNA runs in opposite directions on either side of the strand, right? One side is 5' to 3', which means that the other side has the 3' end over here and the 5' end over here. So on our lagging strand, we end up with lots of little short segments. And that's because when we put down our primer, we can't just keep going this way like we want to, because we have to go towards the 3' side, which is this way. So we start on the 5' side and move towards the 3' side. While we're doing that, the replication fork opens up more space this way. So we put that stuff down, but now we have to jump back. So we can put down a new primer and another short segment of DNA in between where we last started and where we started now. But then we have to jump back. So we have to keep adding these short segments of DNA in the wrong direction, essentially, or like the opposite direction from where the new DNA is coming from. And it's because we can only build in that one way, that 5' to 3'. On the leading strand, it was easy because the 5' is where we started and the 3' is where we're going. On the lagging strand, it's harder because the 3' the ending point is already where we're starting. And so instead, we have to wait for it to open up and then build a little short segment. Let it open up some more, build a little short segment. Let it open up some more, build a little short segment. The short segments are called OKAZAKI fragments because that's the guy who discovered them. I think that's what's on this slide here. Yeah. So the Japanese biochemist Reiji Okazaki was the one who found the DNA building all these short segments and described it in a scientific paper. And so he's the guy that got it named after him. But that's the major difference between our leading strand and our lagging strand. The leading strand can be built in one continuous piece because we start at the 5' side and we're just constantly building towards that 3' side. Easy peasy, no problem. The lagging strand has a problem because it has to be built backwards. So we can start on this section, but then build back to where we started. And then we can put down a second primer and build back towards the first primer. Then we have to put down a third primer and build back towards the second primer. And so on and so forth as the DNA continues to open up right over here. So you guys won't ever have to write like an essay or a paragraph about what are the differences between the leading strand and the lagging strand. But you should be able to tell if somebody describes it to you or if you're shown a picture of it, you should be able to tell which one is the leading versus the lagging strand. And why is it that one strand is able to just continuously add bases, whereas the other one has to form those short fragments? So I'll talk about these guys a little bit more as we move through, and I have a couple good kind of pictures at the end and a video that I'm going to show you guys that shows all this kind of stuff in action. But I know that this stuff is especially tricky. So before we get to that, for now, is there anything that you're still just like totally confused about or totally lost about when it comes to the leading or the lagging strand? Anything that you want me to go back and maybe explain a little bit more or in a little bit different way? Alrighty, I must be doing a pretty okay job with it so far, if nobody has any questions. So again, leading strand, nice, long, easy to make, unbroken piece. Lagging strand, lots of little fragments being built backwards from where we're opening the DNA. So in order to build our DNA and make it that everything ends up as DNA, there are two new enzymes that we're going to talk about. The last two enzymes on your enzyme chart that you're doing for the assignment for today. There is DNA polymerase 1 and DNA polymerase 3. Don't ask me why they're 1 and 3 and not 2. I was not in charge of naming them. I just know what they're called. So DNA polymerase 3 is the first one that I'm going to talk about because it's kind of the biggest, most important one. Polymerase 3 is what is responsible for putting down most of the DNA in DNA replication. So all the parts, all of the leading strand, and all of those OKAZAKI fragments on the lagging strand, all of those are put down by DNA polymerase 3. So whenever you have that RNA primer, polymerase 3 will go right behind it and start filling in the DNA. So it adds the nucleotides, those new bases, on the leading strand and the lagging strand. So polymerase 3 is sort of like the big main workhorse. The way I like to remember the difference between the two is that 3 has a bigger number because it has a bigger job. It's adding most of the new DNA and copying most of the DNA in our DNA replication. Polymerase 1 has kind of a much more specific, specialized job. So you may remember that as we are making our DNA, the little green segments down here represent primers, the RNA primers. And while these primers are super helpful and help us get DNA replication started, they are RNA. They are not DNA. And so eventually, we're going to want to get rid of this RNA and swap it out for DNA, right? We want our final result to be 100% pure DNA, right? We don't want any like 80% DNA and 20% RNA. We don't want any RNA left over at the very end of the process. It's super helpful to start the process. We need it there to get DNA polymerase 3 going, but eventually it needs to go. It needs to get out of town. And that is what DNA polymerase 1 does. So it's a little bit more specific. It's a little bit more specialized than polymerase 3. Polymerase 1 is responsible for essentially cutting out those RNA primers and filling in that short little sequence with DNA. So it gets rid of the RNA and puts down DNA in its place. So on the leading strand, it just has to do this once. It cuts off that starting RNA. It puts down the little DNA. On the lagging strand, it has to go between each one of those OKAZAKI fragments, take out the RNA, replace it with DNA. Go a little bit further, take out the RNA, replace it with DNA. So that is what DNA polymerase 1 is responsible for. It's one of these helper enzymes, and it takes out those little RNA primers that we had and puts DNA up inside of there. Polymerase 3, meanwhile, is just chugging along. It's putting down all the DNA on the leading strand and filling in all of those OKAZAKI fragments on the lagging strand. Again, it's kind of confusing sometimes because they have very similar names and it's 1 and 3 and not 1 and 2 for whatever reason. But like I said, the way that I like to remember it is that polymerase 3 has the bigger number because it has the bigger job. It's doing lots and lots of work, putting down lots and lots of DNA. Polymerase 1 has the smaller number because it has the smaller, more specialized job. All it does all day long is take out those RNA primers and fill it in with DNA. So that's the major difference between polymerase 3 and 1. They both put down DNA, which is why they're named DNA polymerase. It's basically just a fancy science way of saying enzyme that makes DNA, but they do it in slightly different ways, which is why they got these different numbers. So for a little bit kind of clearer view of what's going on, here we have our replication fork. So we have our helicase, which is represented by these kind of 2 ovals down here, moving along, opening up the DNA. Up here, we have the leading strand, and you can tell it's the leading strand because it starts at the 5' side over here and is just continuously in one big long chain making new DNA. So DNA polymerase 3 is going along and building and building and building DNA, continuously adding new DNA bases on top of our original strand. Down here is our lagging strand situation. Now in this case, we have the 5' side is over here, and the 3' side is over here. So we have to put down a primer and then fill in this space by DNA polymerase 3. Jump back, put down a primer, fill in the space, and then once some more bases open up, we'll jump back, put down a primer, and fill in the space. Eventually, DNA polymerase 1 will come by, rip out the primer, put down some new DNA so that it can be all DNA all the time in our lagging strand. All right, so leading and lagging. Constantly being filled in by polymerase 3. Lagging has lots of short fragments being built by polymerase 3. And then all of the little primers being put down are going to be replaced by polymerase 1. All right, so this is basically the same picture we were just looking at, but assuming this works the way that it should, I found a really helpful video that kind of goes through each of these step by step in a really nice sort of 3D way. You're probably not going to be able to hear any of the sound of it, which is fine. It's just a very nice British lady explaining what's going on. But I can point that out to you guys as well. All right, so we've got our DNA strand. Well, let me see how I make this bigger. There you go. We've got our DNA strand, right? We've got our sugars and phosphate groups going all up and down the top and bottom. And then across here are all of our bases, our A's and T's and C's and G's that always line up in that way. So G and C, C and G are always going to be together. T and A and T are always going to be together. So you'll notice that when they made this little animation here, they made sure that T and A are green. So wherever you see them, you'll always see red and green bases together. And C and G are blue and yellow. And so wherever you see them, you'll always see blue and yellow bases together. Just a little reminder from last week. And then again, we are going in opposite directions. So if on the top side, we have the 5' end over here and the 3' end over here, that means that on the bottom side, we have the 3' end over here and the 5' end over here. Always on opposite sides from each other. So the first thing that we need to happen is our buddy Helicase. So Helicase is coming along, unwinding the DNA and splitting it open. So this guy is getting the whole party started. So once we get that DNA opened, we have to have a starting point for our DNA polymerases to come in and start building off of. So that's where Primase comes in. So Primase is going to put down a short segment of RNA to give us a starting point to start building our DNA on. So it takes a little bit of RNA, puts it down on top, and now we have our little RNA primer, and so we're ready to start building some DNA on this side. So now we got DNA polymerase, specifically, in this case, DNA polymerase 3. It's going to come along and it's going to start adding bases. So luckily, we have our leading strand up here at the top. So we're starting on the 5' side, and we're able to build directly in one big long straight line towards the 3' side. So on our leading strand, polymerase 3 can just keep going and going and going and going. Down on the lagging strand, we've got a little bit different story. Helicase is over here, going this way. Going this way. But the way that our DNA is set up is we have to build the DNA, the 5' to 3' side, which is this way, opposite of where the helicase is going. So a little tricky, but nothing our cells can't handle. So we're going to be making a whole bunch of these little OKAZAKI fragments. Primase puts down a little primer. And then we fill it in with DNA, with our polymerase 3. And we're going to do that again and again and again and again. Primer, DNA. So DNA polymerase 1 is also sometimes, depending on sort of the region of the world you're in, called exonuclease, but it's the same thing. So just pretend like this says DNA polymerase 1 right here. I think this might be more of a British term for it. But regardless, it's all about taking off the little RNA pieces that are in here and putting down some DNA. So ligase is another enzyme that we didn't really talk about. It basically just seals all the little places in between all the DNA fragments. And now, at the very end, we end up with 2 copies of DNA that are the exact same as our original copy. And each one has one of the original strands from the parent DNA and one of the new strands that we just built on top of it. All right, so I'm going to go ahead and run this back and let you guys watch it one more time. But this time, I'm not going to pause it, and I'm not going to talk. Or maybe I'll just kind of point out things as we go along, just so that you can really see everything in motion. So we've got our DNA. Helicase comes along and unwinds and opens it up. We then use Primase to put down our little RNA starting point. And then DNA polymerase 3 comes along and starts filling in the DNA bases. It matches them to the original strand. So where we have an A on the original, we'll have a T on the new strand. Where we have a G on the original, we'll have a C on the new strand, and so on and so forth. So when we can put all the DNA bases down in one continuous easy line, it's called the leading strand. The other side of the DNA has an issue of having to constantly make short fragments going in the opposite direction from helicase. It's called the lagging strand. And in this case, we use primase a lot more because we're constantly putting down these RNA primers and then filling in the gap with DNA. So polymerase 3 comes along and fills in the gap. And then we jump back a little bit. Primase puts down a primer. And polymerase 3 comes along and fills in the gap. Finally, polymerase 1 comes in, takes out the RNA, and replaces it with DNA. So now we have two completely identical strands of DNA, where one side of each DNA came from the original parent side, and one side of each DNA is the new DNA that we just put down. All right. So that is the basics of DNA replication. How we make an exact copy of a DNA strand by opening it up and putting down new DNA bases. So before we go on to the very last little piece of this that I want to talk with you guys about, does anybody have any more questions? I know it's kind of a tricky thing, but anything that anybody is still a little bit confused about with DNA replication? Any questions from
No.
All right, Naya's good. Anybody else? Any questions so far?
Fine.
All right, sounds good. So the last thing that I wanted to go over with you guys, kind of a short little tale to this whole story, is that DNA, for the most part, is really, really good about checking itself, making sure that all the correct bases are being paired correctly. And so that polymerase 3 and polymerase 1, as they're putting down the new DNA bases, they're also double checking their work and making sure that the right bases are going to the right bases, making sure that the T always gets paired with an A, or that the G always gets paired with a C. But there is a lot of DNA. There are millions and millions of bases in our DNA that are constantly being copied in order to make new cells. So sometimes there is a mistake. About 1 in a billion nucleotides will get paired incorrectly. So maybe you get a T that pairs up with a C, or you get a G that pairs up with another G. Or sometimes, depending on which bases you can have bases where 2 bases on the same side get paired with each other instead of being paired correctly with the base across from them. So normally when this happens, one of the DNA polymerases will catch it, take out that part, and put it down with new DNA. But about 1 in a billion times, the mistake doesn't get caught, and we end up with a mutation. So there's 3 basic DNA mutations, and luckily, each one, its name is basically exactly what it does. So there is an insertion mutation, which is where an extra nucleotide gets added to the DNA sequence. So it used to be AAT, and now it's AAGT. There is substitution, which is where your one nucleotide gets swapped around for a different one. And then there is deletion, which is essentially just the opposite of insertion, where a nucleotide gets deleted from the sequence. It used to be AAT, and now it's just AT. So I really like this picture because it kind of shows exactly like what I was just talking about. So imagine you have this set of 9 bases down here. It's kind of like our starting point. In an insertion mutation, you get an extra base added. So before, our middle section was ACT, and now there's ACAT. And so now when we make our new DNA, there's going to be a new base. So we have 10 bases instead of the 9 that we started out with. You can also have a substitution mutation where you change around, you take out the C, but you gain an A. So instead of being GAGACT, you end up with GAGAAT. So on the plus side, you have the same amount of bases as you started with, so that's good. But on the downside, one of them is wrong. One of them is different from what it's supposed to be. And then finally, you can have a deletion mutation, which is where we used to have ACT in that middle section right here, but we lose the C and it just becomes a T. So now, instead of having 9 bases, we only have 8. We lose whatever pairing that was going to be in the middle. So depending on where these mutations happen, it could have no effect on you. It might be there are a lot of regions of DNA that actually don't code for anything. They're just kind of called random sections or junk sections of DNA. But if this happens in one of your genes, in one of your sections of DNA that does have the code for building a part of you, then maybe it'll have very little impact, or maybe it'll completely change one of the proteins that you're supposed to be building. Luckily, again, our DNA enzymes are really, really, really good at finding, catching, and fixing DNA mutations so that they hopefully don't end up having an impact on the types of cells and the types of proteins that we're supposed to be making. All right. So that will lead us to the end of our DNA replication notes for today. Does anybody have any final questions about DNA replication or DNA mutations or leading strand, lagging strand, primase, helicase, any of that good stuff. Anything that we talked about today that you still have questions over. All right. So again, your assignment for today, pretty straightforward. Hopefully now you recognize some of these names of these DNA enzymes. And so again, for each one, I just want you to tell me in like 1 or 2 sentences, explain what this enzyme does. How does this enzyme help out with the process of DNA replication? So do that for each one of those 4, submit it, and you will be all set. All right, where's the thing? There we go. One last thing that I wanted to mention before we log off here is that next week, the Excel Center is going to be offering practice STAR tests for people that want to come in and get some extra practice or extra experience taking the STAR test on the computer in one of the rooms where you'll be taking it for real if you are doing that later on in May. So if you are taking either the biology STAR test or the US history STAR test or the algebra STAR test, I believe those are the 3 that we're offering practice for. Next week on Monday, I think it's Monday, Wednesday, and Friday, but we'll check here really quick. Yeah. Okay, yeah. So Monday, Wednesday, and Friday of next week, we'll be offering practice STAR tests for anybody that wants to come in and take a practice version of either the algebra or biology or US history STAR test. It's not like there's going to be a certain one on a certain day. You can come in on any one of those days and take any one of those practice tests. And again, it's a really good way if you want to get experience with taking it on the computer, taking it at the Excel Center in the room where you'll be taking it if you are taking one in May. And so if you want to sign up for a time to come in and do the practice, either let me know or let your life coach know, and we can arrange a time and make sure that there's going to be somebody in the room to help you kind of get set up and take the practice test. And so if you know you're taking the biology, US history, or algebra STAR, and you want to get some practice, let me know and I can help you get scheduled for the time either on Monday, Wednesday, or Friday of next week.
How would you know if you're taking a STAR test?
They may.
They posted a list already of the people who are taking it.
Okay.
You find the list.
A copy of that list to the teachers. The best way is by reaching out to your life coach because they'll know for sure. But I believe I think they also sent me a list of the STAR testers. And so you can also reach out and ask me, and I can see if I can find your name on the list as well. In general, if you're taking a STAR test this term, I'd assume you probably would have heard about it by now. But if you want to double check and make sure, feel free to reach out to me as well.
I was going to ask, do we not take the STAR tests on paper anymore?
Not at the Excel Center. If for people who are not in the Austin area, like we have some students across our different classes that are doing it in different cities, then that might be different for them. But if you're in the Austin area, all of the Excel Center's STAR tests happen on the computer.
And also, I want to ask you, Mr. Michael, there is like any classes that they do tutoring, like in biology?
We do have some Google Classroom set up that have a lot of biology STAR prep kind of questions on it. And you can also reach out to me if you ever want to meet kind of individually or get some extra help when it comes to getting prepared for the biology STAR. So yeah, either me or any of your other teachers, if you want some extra help getting ready for the STAR test, reach out and we will do our best to help you out.
Okay, thank you.
Do you know if they're doing that with history? Because I asked Miss Wendy and she sent me a packet, but I kind of want to do like somebody like to actually talk to me about it instead of me just reading the packet.
I don't know if they have a history like STAR test prep Google Classroom set up. If you want, I can send you have you ever taken a class with Mr. Yambaro before?
No, I don't know who that is.
He's the main history teacher for the Norwood Night campus. So I know with the year going with Miss Wendy, then that's mainly the South campus with who is it? Is it Dr. Fletcher that does it down there? Does a history, I think. I don't know. I think I was remembering correctly. But anyways, I can, if you want me to, I can put you in touch with Mr. Yambaro, who does the main history teacher that I interact with. And you are free to ask him any questions or see if he has any good study materials that he can send your way.
Okay. Yeah, I think I would want to do that.
Okay. Let me write down a quick note. All right, sure thing, no problem. Let's see.